Dear student, I am Dr. 𝗠𝐴𝐲𝐫𝐞, Professor of Biochemistry. In this lecture, I will be explaining you sphingolipids. Let's get into the details. Sphingolipids are composed of long-chain amino alcohol sphingosine. Here is the structure of sphingosine, which has got a long-chain alcohol group, as you see here, and also there is a amide group attached to this long-chain alcohol group. Sphingosine, generally it does not occur in nature, so it has to be synthesized. But how the sphingosine is synthesized? So I am going to explain you that in the next slide. So the purpose of sphingosine is to make another molecule called a ceramide. As you see here, a ceramide, it is a fatty acid which is attached to the amide group of this long chain alcohol. As you can see here, a amide group is further attached with a fatty acid. That makes a ceramide. So ceramide is nothing but it is a sphingosine plus a fatty acid attached to a sphingosine, and that is what is called as a ceramide. And this is what is a little bit introduction to structural aspect of sphingosine and a ceramide. Let's get into synthesis of sphingosine and further how a ceramide is synthesized. Sphingosine, it is formed by a condensation of a hydroxyl containing amino acid, that is serine, and a most common fatty acid that is being synthesized in human cell, and that is a palmitic acid, which is a 16 carbon fatty acid, and it is an activated form here called palmityl-CoA. So the condensation of serine with palmityl-CoA, it is done by a pyridoxal phosphate dependent enzyme called serine palmityl transferase enzyme. Name of the enzyme is serine palmityl transferase. So as it is there in the name, or this enzyme is going to condense hydroxyl group containing amino acid serine with the palmitic acid in its active form called palmityl-CoA. So now with that we get 3-keto dihydrosphingosine. This is how a sphingosine is made. Now to this sphingosine, a fatty acid is attached. So ceramide, as I said earlier, ceramide is a long chain fatty acid amide derivative of a sphingosine. Alright, so as you see here, to sphingosine, especially to the amide group, there is an attachment of a fatty acid, and that is how a ceramide is made. So now let's see how a ceramide is converted into a sphingomyelin. So because ceramide is a core structure of natural sphingolipids. Note that a ceramide is the core structure of natural sphingolipids, and sphingolipids are nearly present in all body tissues, and also it is present in the blood. Now the highest concentration of sphingolipids are found in the white matter of central nervous system. Remember that. So white matter of brain contains highest amount of sphingolipids. And also different types of sphingolipids are the components of the plasma membrane of all our cells. Now let's look at how a sphingomyelin is synthesized. Now let's move on to see how a sphingomyelin is synthesized. Sphingomyelin, it is a ceramide with phosphocholine. That's what is sphingomyelin. So very simplified way of writing a sphingomyelin is it is a ceramide plus phosphocholine. That means choline with a phosphate attached. Now ceramide which is attached with phosphocholine, it is synthesized by sphingomyelin synthase enzyme, where a phospholipid called phosphatidylcholine, it is going to donate its phosphocholine moiety to the ceramide here and make sphingomyelin. As you see in the figure, here is the ceramide which is going to take a phosphatidylcholine part of phosphocholine. So phosphocholine is there in phosphatidylcholine. Only the phosphate and the choline moiety is taken and we get this sphingomyelin, and the rest of the molecule in phosphatidylcholine is released as diacylglycerol. This is how sphingomyelin is made and done by sphingomyelin synthase enzyme. Now, as you see in sphingomyelin, so there is a fatty acid attached and there is a sphingosine alcohol and there is phosphate here and also there is choline. These are the components of sphingomyelin. So if you break it, so this part here, it is sphingosine. Here is the fatty acid attached to sphingosine, and here is the phosphate, and here is the choline. Okay, so that means you can break it down into 4 components there. One is a sphingosine, which is an alcohol. Other is a fatty acid. So sphingosine plus fatty acid, it is what is a ceramide. And then there is phosphate and choline, so phosphocholine. So otherwise it is ceramide plus phosphocholine. That is what is a sphingomyelin. You should be able to identify the structure of sphingomyelin. So here in this slide, I am explaining the little more information on what I have said before. All sphingolipids contain a sphingosine, a long chain amino alcohol. Sphingosine does not occur naturally, hence it needs to be synthesized. Sphingosine, it is formed from condensation of serine with palmityl-CoA done by an enzyme serine palmityl transferase, and that enzyme needs pyridoxal phosphate. And ceramide, it's a long chain fatty acid amide derivative of sphingosine. And ceramide is a core structure of all natural sphingolipids. And sphingolipids are present in all body tissues. And the highest concentration of sphingolipids are found in white matter of the brain. And different type of sphingolipids are components of our plasma membrane. And the structure of sphingomyelin, just to recap, and it's important for you to identify the structure of sphingomyelin. So sphingomyelin, it is, as I said, it is a ceramide plus phosphocholine, and you can break that sphingomyelin into 4 different components. That is, it is sphingosine plus fatty acid. Sphingosine plus fatty acid together is a ceramide, and then phosphocholine, so phosphoric acid and choline. So here sphingosine, fatty acid, phosphoric acid and choline, that's what is a sphingomyelin, or you can say ceramide plus phosphatidylcholine. So this is how you can remember a sphingomyelin structure there, or the component. Alright, know that a sphingomyelin is the only sphingolipid that is a phospholipid. And also a sphingomyelin is a major structural lipid that is present in a cell membrane of central nervous system. And the most common type of fatty acids that are found in sphingomyelin are either palmitic acid, which contain 16 carbon, stearic acid, which contains 18 carbon, lignoceric acid, a 24 carbon fatty acid, or nervonic acid, which is a 24 carbon monounsaturated fatty acid. So meaning, since sphingomyelin is present in the highest concentration, in the brain. And note that the type of fatty acids that you are going to find in sphingomyelin, some of them are very long chain fatty acids like lignoceric acid and nervonic acid. So, when the sphingomyelin undergoes degradation, these very long chain fatty acids need to be oxidized, and they will be oxidized in the peroxisomes. That's something that you need to remember. Note that in the white matter, the sphingomyelin, it has higher content of like lignoceric acid and nervonic acid, whereas that in the grey matter, it contains largely long chain fatty acid, that is a stearic acid. So, note the difference there. Okay. So, in white matter, very long chain fatty acids are found attached to sphingomyelin, whereas in the grey matter, it is only long chain fatty acid like stearic acid. And excessive accumulation of sphingomyelin, it is seen in a disease called Niemann-Pick disease. Now let's move on to see glycosphingolipids. Glycosphingolipids, they contain ceramide attached with a monosaccharide or oligosaccharide. So it is a simple structure, a ceramide attached with a carbohydrate. That is what is glycosphingolipid. Glycosphingolipids are constituents of outer leaflet of plasma membranes, and also these are important in cell adhesion and recognition. Now coming to cerebrosides. Cerebrosides these are the ceramides that are attached with a monosaccharide. As you see here in the figure, a ceramide plus a glucose. It is a glucocerebroside or a glucosyl ceramide. That's how we can say it as. Or if it is a galactose attached to ceramide, it is galactosyl ceramide or galactocerebroside. So, cerebroside is a ceramide plus monosaccharide. If it is a glucose, it is glucocerebroside or glucosyl ceramide. If it is galactose, it is galactocerebroside or galactosyl ceramide. This is how we can refer to these molecules. Now note that nucleotide sugars, that is UDP glucose, UDP galactose, these are the molecules that enters into formation of cerebrosides, because the ceramide is the basic structure and monosaccharide that is going into the reaction. So always remember, the monosaccharides or the modified sugars, they all enter into reaction in their nucleotide form. And here for glucose and galactose, it is the uridine diphosphate, that is a nucleotide which is taking monosaccharides to attach it to ceramide to make a glucosyl ceramide or galactosyl ceramide. So here glucose and galactose, they go in as UDP glucose or UDP galactose. These are simply referred as nucleotide sugars. Now, among glucocerebroside and galactocerebroside is the most common cerebroside found in the plasma membrane, especially it is rich in brain. And note that increased amounts of galactocerebroside accumulates in the white matter in a disease called Krabbe disease. And Krabbe's disease is also referred as globoid leukodystrophy. And glucocerebroside is a minor component of plasma membrane. It serves primarily as an intermediate in the synthesis and degradation of the more complex glycosphingolipids. And increased quantities of glucocerebroside is found to be accumulated in the spleen and liver of patients suffering from Gaucher's disease. We are going to go into details of these disorders, but at this point in time, know the name of the disease and also what kind of molecule is accumulated in the tissue of these patients. So, in Krabbe's disease, galactocerebroside is accumulated. You know, galactocerebroside is also called as galactosyl ceramide. Don't get confused. Cerebroside ceramide. So galactocerebrosid also referred as galactosylceramide. Alright. So similarly in Gaucher's disease, glucocerebrosid or glucosylceramide. This is the molecule that is accumulating there. Now let's move on to see globosides. Globosides are the ceramides which are attached with two or more monosaccharides, that is the oligosaccharide chain that is attached. And the monosaccharide units are usually galactose, glucose or N-acetyl galactosamine. So globosides are neutral compounds and they contain no free amino group. Examples for globosides, we have lactosylceramide, that is glucose plus galactose attached to ceramide is referred as lactosylceramide, as you see here in the figure. So ceramide is there, to that glucose and galactose is attached. So glucose plus galactose is lactose, as you know. That's why it is called as lactosylceramide. And this lactosylceramide is a component of erythrocyte membrane. Now another prominent globoside is ceramide trihexoside, and this ceramide trihexoside is also called as ceramide galactosylactoside. And ceramide trihexoside, some books will mention it as globotriazoylceramide. This is another name for ceramide trihexoside, globotriazoylceramide. So the structure of globotriazoylceramide or trihexosylceramide is written here. Trihexosylceramide, as you can see, there is a ceramide which is the basic structure, and then three hexoses are attached to it: glucose, galactose and galactose. Three hexoses, all of these, like glucose and galactose are, as you know, 6 carbon containing monosaccharides, hexoses. So three hexoses are attached to ceramide, that's why it is called as trihexosylceramide. So ceramide trihexoside. So make sure you remember the name here. The reason for that is, in a disease called Fabry's disease, there will be accumulation of ceramide trihexoside. That's why you need to remember the name here. So ceramide trihexoside, also referred as globotriazoylceramide, which is accumulated in Fabry's disease. Now let's move on to see what is sulfatides. Sulfatides these are sulfuric acid esters of galactocerebroside. And sulfatides are found predominantly in the nervous tissue. And the sulfate donor in sulfatide formation is a storage form of sulfate in our cells, and that is PAPS. And the PAPS here is 3' phosphoadenosine 5' phosphosulfate, which is also referred as active sulfate. Remember this point, very important. So the sulfate donor in our cellular reaction is predominantly, it is PAPS, that is 3' phosphoadenosine 5' phosphosulfate, which is referred as an active sulfate. So here in the figure we have ceramide, basic structure, UDP galactose is coming into the reaction. So we got like galactosylceramide, that's how galactosylceramide is made. So galactosylceramide is a type of cerebroside. Okay, and then PAPS is giving sulfate, so it becomes sulfogalactosylceramide, which we just refer it as sulfatide. Now galactocerebroside 3-sulfate, that is sulfogalactosylceramide, is the major sulfolipid in the brain, and it accounts for 15% of the lipids of white matter. So that's a significant amount there. And increased quantities of sulfatide is accumulated in a disease called metachromatic leukodystrophy. So here are some of the structures of gangliosides, not really structures per se, it is just how a composition or the components in different type of gangliosides are seen. So we have a GM1 ganglioside. So we have a GM1 ganglioside, GM2 ganglioside and GM3 ganglioside. Let's look at the difference in these three types of gangliosides. As you know, ceramide is the basic structure, so all of them contain ceramide. So ceramide is there. So there is a ceramide. In all of them there is ceramide. And then all of them contains glucose. All of them contain glucose. Then all of them contain galactose. That is common. So that means ceramide, glucose, galactose. All gangliosides contain these. And then GM3 ganglioside, to its galactose there is an attachment of sialic acid, that is N-acetylnuraminic acid. That's the GM3 ganglioside. So it is ceramide, glucose, galactose, N-acetylnuraminic acid. That's GM3. And then look at GM2 ganglioside. GM2 ganglioside has ceramide, glucose, galactose, N-acetylnuraminic acid. Along with that, there is an attachment of N-acetylgalactosamine, that is GalNAc. That's an additional attachment to galactose there. That makes it little more complex than GM3. That is what is GM2. And then the GM1, you see there is glucose, galactose, N-acetylnuraminic acid. Now you can say this is found in all gangliosides, GM1, 2, 3. Common thing here is ceramide, glucose, galactose, N-acetylnuraminic acid. All three of them has that. And the GM2 has an additional N-acetylgalactosamine. Now the GM1, apart from having that N-acetylgalactosamine, it also has one more galactose attached. So as you go from GM3 all the way to GM1, the complexity is increasing, or little more components in the structure is increasing. So this is what is the difference between three gangliosides, GM3, GM2 and GM1 ganglioside. Now note that why these molecules are referred as GM1, 2 and 3. Let me just explain you that particular aspect. It is just to know why these GM and 1, 2, 3. So G here indicates ganglioside. That's what is G indicates. Okay, and M indicates monosalic acid. Means attached with one sialic acid. As you see here, in all GM1, 2, 3, so there is only one N-acetylnuraminic acid is added. Only one sialic acid is present. That is what is mono. So ganglioside mono. So as I said now, G for ganglioside, M for mono, that is one sialic acid. Now let me explain you why number 1, 2, 3 is given here. So when these gangliosides are separated in paper chromatography, and the sample is applied on one side, and if the sample runs from left to right in the paper chromatography, so the molecule which is simple, like littlest component, like smaller, that will move faster. That means GM3 ganglioside will move faster, that will be here, and then GM2 will be moving behind that GM3, and then GM1 do not move much. So that means we will give the numbers as number 1 position, number 2 and number 3. So the bigger molecule not moving much, that is GM1 ganglioside, and then the next simpler molecule is GM2 ganglioside, and the smallest one here is GM3. So that 1, 2, 3 is the how they move in the paper chromatography when they are separated based on the complexity. That's what is the numbering. So G for ganglio, M for monosalic acid, 1, 2, 3 is the how complex they are. So 1 is most complex and 3 is the least complex based on the composition. That's how it is. Now, gangliosides are predominantly found in ganglion cells of the central nervous system, and that's why they are called as ganglio, because they are richly available in ganglions. And gangliosides are synthesized from the ceramide by a stepwise addition of activated sugars, and they are UDP glucose, UDP galactose, and then we have sialic acid and N-acetyl neuraminic acid. Most of the enzymes transferring these sugars from nucleotide sugars are specific glycosyl transferases, which are all found in the Golgi apparatus. That means ganglioside formation is going on in the Golgi apparatus. Remember that. And increased quantities of gangliosides accumulate in tissues in GM1 gangliosidosis disorder and GM2 gangliosidosis. And GM2 gangliosidosis is also referred as Tay-Sachs disease. So the figure here, it is showing you how a ganglioside is made. So it starts from ceramide, UDP glucose getting into the reaction and added, only glucose is added, UDP is released by specific glucosyl transferase. So we get glucosyl ceramide, which is called as glucocerebroside. So we have made cerebroside there. And then to this, a galactose is being added. So it is coming in as UDP galactose. So ceramide glucose galactose, that is lactosyl ceramide, and lactosyl ceramide is a globoside. So as we go here, let me explain you that concept. So we have cerebroside, we have made cerebroside. As we are going towards ganglioside, so there is cerebroside formation, and then cerebroside is converted to globoside, and then there is an addition of N-acetyl neuraminic acid, that is a sialic acid, and with this sialic acid addition we got ganglioside now. Now let us understand what are the differences between cerebroside, globoside and ganglioside. So cerebrosides are ceramide plus monosaccharide. It can be glucose or galactose. If it is glucose, it is glucosyl ceramide, or if it is galactose, it is galactosyl ceramide. And then to this cerebroside, if one more glucose or galactose is added, so it becomes globoside. So globoside is, it is ceramide plus disaccharide or oligosaccharide. And then to this globoside, if a N-acetyl neuraminic acid is added, that is sialic acid is added, right? So that becomes a ganglioside. So the ganglioside that is made here in the pathway, it is GM3 ganglioside, a simplest ganglioside. Now to this GM3 ganglioside, N-acetyl galactosamine is added additionally, and that becomes GM2 ganglioside, little more complex than GM3 ganglioside. And then to this GM2 ganglioside, a galactose molecule is added, and that becomes GM1 ganglioside. So this is how a gangliosides are synthesized from a ceramide molecule in a stepwise manner. Remember that. Now let's look at functions of gangliosides. Gangliosides are structural component of plasma membrane. Nervous tissue contains a maximum amount of glycosphingolipids. Sulfogalactosides are important constituents of myelin, and gangliosides and galactosides are abundant in grey matter of the brain. So they are all located in the outer layer of the plasma membrane, and thereby they interact with the extracellular environment. So you got to pay attention to some of the details here. So the first thing is the nervous tissue. That's where we get these glycosphingolipids, basically the sphingolipids, which are added with the carbohydrate, are richly available in nervous tissue. The second point that you need to remember is that is the sulfogalactocerebroside. See, the galactosyl ceramide or galactocerebroside, which is sulfated, and that is the important constituent of the myelin sheath. Remember that. And the third thing is gangliosides and galactocerebrocytes are abundant in the gray matter of the brain. So these are 3 points that you must remember here. Now the second function is ganglioside. They function in cell-cell interaction and antigenicity. Now the glycosphingolipids, they play a key role in cell-cell interaction, growth and development. They have been identified as tumor antigens, blood group antigens and embryonic antigens specific for particular stages of fetal development. That's where glycosphingolipids come in. Now the other function of ganglioside is they can act as a membrane receptors. So the gangliosides act as receptors for toxic agents and some pathogens like Vibrio cholera, influenza virus, tetanus toxin. There will be changes in the glycosphingolipid composition of membrane in various malignant disorders also. So these are some of the functions of gangliosides. So in this lecture, I have explained you the chemistry of sphingolipids. So how a ceramide is synthesized from a very basic structure, that is sphingosine. A sphingosine is nothing but it is a serine plus palmitoyl-CoA, and to that serine and palmitoyl-CoA, if a fatty acid is attached, that makes ceramide, and that ceramide to this, if a phosphocholine is attached, it becomes a sphingomyelin, and like that we have seen different structures after that sphingomyelin. We have seen like cerebrocytes, globocytes, gangliosides, different types of gangliosides, and also the location, composition and the functions of all these sphingolipid molecule. So that's all about sphingolipid chemistry. Thank you.